Okay, so today we will be having a neuroscience presentation. Keerthi and Shambhavi helped with the slideshows, and thank you guys for volunteering for on the spreadsheet for this topic. Yeah, so what is neuroplasticity? So neuroplasticity can be viewed as a general umbrella term referring to the brain's ability to modify, change, and adapt both structure and function throughout life, right? Um, yeah, throughout life. And so it actually depends on experiences. Depending on our experiences, different pathways form or fall dormant or are created or discarded. So this is actually a really important concept. So let's watch a little short video here.
Not so
Sorry. Can you guys hear it?
long ago.
Yeah, a little, but it's a kind of glitchy, so you might want to share your computer sound.
Yeah. I don't know why my Zoom controls just disappeared. Oh, it's right here. It takes a long time to load. Sorry, guys. So I'm going to share my computer sound.
Many scientists believe that the brain did not change after childhood, that it was hardwired and fixed by the time we became adults. But recent advances in only the last decade now tell us that this is simply not true. The brain can and does change throughout our lives. It is adaptable, like plastic. Hence, neuroscientists call this neuroplasticity. How does neuroplasticity work? If you think of your brain as a dynamic, connected power grid, there are billions of pathways or roads lighting up every time you think, feel, or do something. Some of these roads are well-travelled. These are our habits, our established ways of thinking, feeling, and doing. Every time we think in a certain way, practice a particular task, or feel a specific emotion, we strengthen this road. It becomes easier for our brains to travel this pathway. Say we think about something differently, learn a new task, or choose a different emotion. We start carving out a new road. If we keep travelling that road, our brains begin to use this pathway more, and this new way of thinking, feeling, or doing becomes second nature. The old pathway gets used less and less and weakens. This process of rewiring your brain by forming new connections and weakening old ones is neuroplasticity in action. The good news is that we all have the ability to learn and change by rewiring our brains. If you have ever changed a bad habit or thought about something differently, you have carved a new pathway in your brain and experienced neuroplasticity firsthand. With repeated and directed attention towards your desired change, you can rewire your brain.
All right. Oops, sorry. Let's not restart the video. All right. So, what's the origin behind neuroplasticity? So, Santiago Ramon y Cajal, the father of neuroscience, used the term neuronal plasticity to describe non-pathological changes in the structure of adult brains. He referred to the term in his findings of degeneration and regeneration in the central nervous system after adulthood. And he's actually the person on the left, the picture on the left. And Carl Lashley, an American psychologist, conducted experiments on rhesus monkeys that demonstrated changes in neuronal pathways, which he concluded as evidence of neuroplasticity. So he proved in his experiments that it actually existed. There are four main types of neuroplasticity adaptations. The first one is neurogenesis, then synaptogenesis, long-term potentiation, and long-term depression. And so neurogenesis is just the creation of new neurons, synaptogenesis, new neural connections. Then long-term potentiation is a short phrase for that is fired together, wired together. So the neurons that fire together actually wire together when you strengthen synapses through recurring activities. That pathway strengthens. And so that was actually seen in the video with the arrows. And the more you actually went along the pathways, the more you would strengthen it. Then the last one is long-term depression, which is weakening of synapses that aren't being used anymore. So that's just use it or lose it. Previously, it was believed that only children can regenerate neurons. But now new research finds that neurogenesis occurs at high rates in young brains and can still occur in the adult brain, though not at such high rates, until around 10th decade of life, which is when you're like 100. Yeah. And long-term depression is actually associated with memory and motor learning. So its harmful effects can easily be seen with Alzheimer patients, because you'll lose that ability. Like Alzheimer patients lose the ability to remember things. And so you just use it and lose it, or use it or lose it. Yeah. Let's see. So there are 4 different types of neuroplasticity, and there's actually a diagram in the upper right corner for each. So the first one is homologous area adaptation. It occurs during the early critical period of development. And if a particular brain module becomes affected, normal operations have the ability to shift from the affected brain region to an unaffected brain area. An example of this is when the right parietal lobe becomes damaged early in life and the left parietal lobe takes over visuospatial functions at the cost of impaired arithmetical functions. But this actually isn't too bad for us because the timing is crucial here in the development of kids. As kids, we actually learn to navigate physical space before learning arithmetic. So that cost there isn't too high, and we actually can learn it back again. Compensatory masquerade is the second one. It's when the brain figures out an alternative strategy for carrying out a task, when the initial strategy or the initial route that you traveled through is impaired. A person who suffers some form of brain trauma and impaired spatial sense would resort to the strategy of spatial navigation, or another strategy for spatial navigation, like memorizing landmarks. So you use your memory instead of your spatial sense to actually get to places. And cross-model reassignment is a third one. It introduces new inputs into a brain area deprived of its original main inputs. And a classic example of this is the adult's ability to have touch input redirected to the visual cortex in the occipital lobe, even if they were blind since birth. And the reason why this works is because neurons can communicate with each other in the same language of electrochemical impulses, regardless of whether or not they have true vision. The visual cortices of blind people can still carry out cognitive functions of creating representations of the physical world, but those representations come from an input of another sense, like touch. It's change in the actual functional assignment of a local brain region. And the last type of neuroplasticity is map expansion, which allows the local brain regions to be flexible in performing a type of function. And map expansion is also seen in a phenomenon that Keerthi will explain called phantom limb syndrome. Yeah.
Okay. So I'm gonna go 3 main types of neuroplasticity works. So the first is John Cass, and the second is Phantom Limbs, and the third is work by Michael Merzenich. So John Cass, so basically the experiment that he made it's called large-scale reorganization in the somatosensory cortex and thalamus after sensory loss in macaque monkeys. So in this case, the macaque monkeys that Cass and his team used have a spinal cord injury called unilateral dorsal column lesion, and it's basically a injury in the spinal cord. And as shown in the bottom right image, the spinal cord is directly connected with the brain, obviously. And so it connects the primary somatosensory cortex and the thalamus. And particularly in the thalamus, it connects the VP, or the ventroposterior nucleus. And so the somatosensory cortex and the VP get information about senses from all over the body. And so a damage in the spinal cord would basically damage the VP and the somatosensory cortex. And what Cass and his team expected was that because of this injury, the somatosensory cortex and the VP should be damaged like completely because the spinal cord is injured. But what happened really was that the connectivity after these injuries showed regrowth, even though they were adult macaques. And so usually what we think is that in children, neuroplasticity is much greater. but it actually proved otherwise, and that even in adults, there was neuroplasticity because of the regrowth of neurons, regardless of the damage that happened. Next is phantom limbs. So phantom limb is basically a term coined for a limb that is amputated or missing. So phantom limb pain is basically when you feel pain even though there's nothing there. And that can be seen by the image on the top right corner. And so like the person feels pain on his leg even though his leg isn't there. And so the mirror box is a concept that was created by V.S. Ramchandran, who is a professor at UC San Diego. And the picture on the right bottom corner depicts the mirror box. And so the mirror box is basically where a person who has an amputated hand or something, if they put their intact hand on one side and then their amputated hand on the other side, they assume that their amputated hand is actually there because they look on the mirror. And so I don't know if you can like tell by the image, but that's actually what's happening. So what Ramchandran and his team said was that if someone has an arm that's amputated, there's really no reason for that arm. So the brain rewires itself so that the part controlling that amputated limb is covered up by surrounding parts that would be more useful. So the first image on the top row depicts this. So basically after the amputated arm, the section, it's in orange, it's circled on the first image of the brain. So that's basically the part of the brain like responsible for controlling that arm. And then the brain basically transforms and it sort of contracts. So the blue region is where the brain contracts in on itself, and it basically takes out the area that's controlling the arm because there's no reason for it. And instead the outer regions take over. And that can be seen by like the actual brain itself in, I don't know if you can tell by it, like the red region is now the purple and then the purple's like contracting. And so, yeah. And then there's another thing called maladaptive plasticity, and it was created by Herta Floor. And it basically said that cortical remapping occurs because of phantom limb pain. So this is different from what Ramchandran said because he said that cortical remapping occurs because of phantom limbs, but it's actually not just phantom limbs, it's pain. So if you feel pain on those phantom limbs, that's when cortical remapping occurs. And then lastly, there's Lorimer Moseley and Peter Brugger, and they basically executed an experiment where their patients kept imagining that their limbs moved in very like weird and impossible ways. And that indicated that the brain was changing since there needed to be motor movements in order to execute these commands. And so one thing you have to keep in mind is like this hand was amputated. So the hand wasn't physically there moving like backward or something. It wasn't there, but they were thinking it was moving in weird ways. So this sort of indicates that their brain was thinking in a different way than normal humans would, meaning that there had to be some kind of change, right? Like a regrowth in their brain. So that basically indicates neuroplasticity as well. Yeah, and then the next slide is about Michael Merzenich. So Michael Merzenich basically did, along with Clinton Woosley, they did an experiment with monkeys where they cut the peripheral nerve of the monkeys. And a peripheral nerve is basically a nerve that relays information between your brain and the rest of the body. So they created a hand map, and a hand map is just a map of your hand of the monkey before they cut the peripheral nerve and after they cut it. So they expected the map to be very jumbled up since the peripheral nerve, it relays information, right? So if you cut it off, obviously the brain should be like different, very jumbled up. However, after they sewed the peripheral nerve back together, the surprising thing was that the brain was completely normal. And the hand map, the hand map basically is like the portion of the brain. It's not actually like a complete hand map or anything. It's the brain that controls the hand. And so the hand map shows on the image, I tried to circle it, but so the blue region, basically the sections 2 and 3, essentially expanded rapidly over sections 1 and 4. So they basically took over. And again, this is like what we saw previously in phantom limbs. It's basically like map expansion, where certain parts of the map like expand onto other parts because a certain limb or something is taken out. So in this case, like a finger was taken out, and because the finger was taken out, certain parts of the hand map weren't expanded because some parts weren't used anymore. And yeah, and this indicates that there was plasticity because of the regrowth and expansion. And yeah.
Okay, so what does neuroplasticity look like? There's actually two types. I'm just going to go briefly over it. But basically, when you experience something new, like learning a new language or playing an instrument, the neurons in your brain form new connections. And so the first one is like axonal rerouting. If you remember the TEDx video we watched in the Alzheimer's presentation, the speaker, her name was Lisa Genova, had said that if you still remember who she is, even if you've forgotten her name, you can still remember it based on remembering that she's written a book called Still Alice. You still have a way to reroute the wreckage as shown in the diagram on the left. And then the diagram on the right is just sprouting. So we'll have a short video. I'm actually going to skip a few parts to the end, and then just Shambhavi will explain that diagram. So just very quickly, I'm going to show. Let's see. So this girl, let me see if I'm sharing my audio again. Oh yes. So this girl has epilepsy, has a syndrome called Rasmussen syndrome, and then so I'll show you a little bit right here. It's really sad what she experiences. And so she had to do a right hemispherectomy, which is where they cut out half of her right brain. And I'm just going to show you that little clip where she's actually training to walk again. And this shows neuroplasticity.
Left her with a hemiplegia, a stroke-like effect on her left-hand side.
Girl, look how good you're walking.
She's now moved to the Kennedy Krieger Institute, a specialist rehabilitation center, where she'll undergo 3 weeks of intensive therapy. The remaining side of her brain must learn to control both sides of her body.
Can you kick my hand, Cameron, with this foot? Kick me. Good job. Kick me with this foot. Come on, try. You can do it real hard. Oh, I saw your foot move. There it goes. There it goes. Good try.
Yeah. So basically, let me try to exit out of this. What that really means is like she was trying to rewire her brain after the surgery because her right hemisphere was completely gone, and the right hemisphere controls the left side of her body, and that's why you saw that she couldn't actually move her left knee. And then now Shambhavi will just go back to this diagram. Oh, sorry. Go back to the diagram and just talk about the sprouting.
Okay, so if you go back to the axonal sprouting diagram, if you take neuron A as Cameron's right brain, you're going to take neuron A as Cameron's right brain. And this is because the connections from her right hemisphere that were severed in the hemispherectomy, axons and dendrites from the left hemisphere. Neuron B in the diagram was able to take over the motor controls that Cameron lost, and therefore she was able to learn how to walk again.
Yeah, it's really sad how like she has at such a young age she has to experience that. But this example really shows neuroplasticity, and I really encourage you guys to actually watch this video that we'll release with the slides. Yeah.
Okay, so I'm briefly just going to go over 2 examples of neuroplasticity. So the first one is the sea monken, and they're a tribe of sea nomads that live in the island Archipelagos along the west coast of Thailand. And in 1999, Anna Gilson from the University of Lund in Sweden, she was studying the unique characteristics of the Moken tribe. So she set up an experiment to test just how good the children's underwater vision really was. And so the test was just that the kids had to dive underwater and place their heads into a panel, and from there they could see a card displaying either vertical or horizontal lines. Once they had stared at the card, they would have to come back up to the surface to report which direction the lines traveled. And each time they dived down, the lines would get thinner, making the task harder. And it turned out that the Moken children were able to see twice as well as European children who performed the same experiments experiment at a later date. But Gilson wondered whether the Moken children had a genetic anomaly to thank for their ability to see underwater or whether it was just down to practice. So, to find out, she asked a group of European children out on holiday in Thailand and a group of children in Sweden to take part in the training sessions in which they dived underwater and tried to work out the direction of lines on the card. So, basically, the same experiment that the Thailand children did. And then after 11 sessions across 1 month, both groups had attained the same underwater uh, acuity as the Moken children, which goes down to show that this is also a work of neuroplasticity, and that after lots of practice, the Moken children and the children in Europe were also able to see underwater. And also, the way they could see underwater was that they could constrict their pupils by 22%, which means they made their pupils smaller. And this is actually a similar thing to what dolphins and seals do. And adults and elders weren't able to do this because they lost their lens flexibility. So this is just one example. And then the next example is the London cabbies. So these are taxi drivers in London. And so basically, so the area of the brain that was in so most of these taxi drivers were men, but that didn't really affect anything. But the area of the brain that was impacted was the hippocampus. And so the thing that what happened was that a lot of gray matter was taken and their hippocampus had grown because after 3 to 4 years of learning all the streets and routes of London, they had a very detailed spatial map of the city in each of their heads. And so this made their hippocampus larger than a bus driver's. So if you look at the diagram here to the right, you can, I think diagram A is the bus drivers and diagram B is the taxi drivers. And there's actually I think switch that around. So in A, there's an increase in the hippocampus, and this is because their brain had expanded to contain all the gray matter that the drivers had learned from learning the mental atlas of the London streets. Yeah. And then the downside to having a big hippocampus is that when the cabbies retire and stop using their spatial mapping so regularly, the hippocampus actually starts to shrink back to normal. And it's like a muscle that shrinks if you don't use it. So when the gray matter also kind of just disappears too, which is a big downside. And so researchers also found that the cabbies were worse at remembering things based on visual information and had worse short-term memories. But the plus side is that these cabbie drivers were pretty rare, and their knowledge was known as the knowledge because it was pretty advanced to learn uh, the whole like map of the streets, and it took 3 to 4 years to complete. So the brain had grown to adapt over time. So that's also another example of neuroplasticity.
Yeah. So I'm just going to go over this briefly. It's just how to increase neuroplasticity. If you have new experiences, it increases new neural pathways, and same with recurring practice. Heavy repetition of a certain like activity strengthens neural connection. And these are just exercises that you guys can look at afterwards. And how can you use neuroplasticity to your advantage? So really, when you like set a goal for yourself, when you set little small goals and then reach towards it, you can actually increase your dopamine because you find that you reach that little goal and then you are more motivated to reach the second little goal. And so with that, you can increase your neuroplasticity because the more you fire the neurons that actually increase the dopamine, the more they'll wire together. And so the more you find satisfaction in doing a specific thing, like training for a marathon, the more you'd be able to actually do it in the future. And so we can talk about this later. It's actually in the speaker notes if you guys want to see it later. But right now, I am going to start the Kahoot.